Hello everyone. Welcome to Life, the Science. Today I am going to tell you guys one of the most useful techniques in molecular biology, PCR or polymerase chain reaction. So, let's get started. PCR or polymerase chain reaction is a versatile method for in vitro amplification of a specific target DNA molecule within a long stretch of DNA sequence. The technique was formulated by American biochemist Kary Mullis in 1985, for which he was awarded Nobel Prize in 1993. Now, let me tell you what are the major components of this technique, or what are the components need to be added for the amplification. The first one is primers. We need to create two primers, one is forward primer and one is reverse primer. As, in case of DNA replication, short RNA primers bind to each strands of the DNA for the initiation of replication, just like that primers are needed here. As per experiment, optimal length of primers for PCR is 18 to 20 nucleotides. Second, DNTPs. Third, divalent cation, usually Mg2+, which acts as cofactor for DNA polymerase. Fourth, buffer, which helps in maintaining pH of the solution. Fifth, template DNA or the target DNA. And the sixth one, the most important, thermostable DNA polymerase. Why thermostable? We are coming to this in the next part of the lesson. This is a reaction cycle of PCR which is generally called thermal cycle. We can divide the whole cycle in three steps: denaturation, annealing, and extension. Before starting DNA replication, as helicase separates two strands by breaking the hydrogen bonds between base pairs. Like that, in PCR, we use high temperature to separate the two strands. The required temperature is 93 to 95 degrees centigrade here. At this high temperature, normal DNA polymerase is not stable and loses its properties, so we need to use thermostable DNA polymerase. In general, thermostable DNA polymerase is used. Next step is primer annealing, which occurs in 55 to 60 degrees centigrade. And the next is extension of primers by adding DNTPs, catalyzed by thermostable polymerase. The cycle is repeated based on the requirements, like 20 to 30 cycles. After each cycle, the number of templates doubles. This number can be calculated by applying the following formula. Final is equal to initial multiplied by 2 to the power number of cycle. So that, if 1 starts with a single double-stranded DNA molecule after 20 cycles, the number of molecules synthesized by the PCR is 1 cross 10 to the power 6. And, after 30 cycles, the number increases to 1 cross 10 to the power 9. So, that's all for today. Thank you. Like, subscribe, and share.